Good morning. So today we are going to talk about meiosis. So what happens during meiosis and what is meiosis? Let me tell you. Meiosis is a reductional division which can be seen in sexually reproducing organism. If I talk about human being, especially human being, so what happens during gametogenesis? During gametogenesis, it occurs just to maintain the ploidy. Okay, so the formation of gamete takes place through meiosis. If I also talk about in fungi and in bryophytes, what happens? You can see the meiosis there in zygote. That type of meiosis is called zygotic meiosis. I hope you know that. That is called zygotic meiosis. What happens in them? Their zygote is diploid while their parents are haploid. So, in order to maintain the ploidy, the zygote goes through meiosis to maintain the ploidy. While in sexually reproducing organism, what happens? The parents now see this is why do we inherit? Why do we inherit 23 chromosome from father, 23 chromosome from mother? Because we are 46. We have 46 chromosome, and to maintain that 46 number, what happens in father and in mother during gametogenesis? Meiosis occur. Sorry, meiosis occur, which decreases the number of chromosome by 2. That is, if our father has 46 chromosome and our mother has 46 chromosome, so the number of chromosomes will get half after meiosis. The 46 will become 23, 46 will become 23, and after fertilization, the number of chromosome will get restored. That will be 46 in a baby. Okay, so let's talk about meiosis in detail. Meiosis occur in two stages, that is meiosis first and meiosis second. If we talk about meiosis first and meiosis second, in between them there is stage, interkinesis stage, and before meiosis first there is G1 phase, S phase, and G2 phase. Okay, but I want to tell you that between meiosis first and between meiosis second there is no S phase. That means the duplication of DNA, that is the number of DNA gets doubled only one time during meiosis. Okay, so DNA get doubled here only one time. Okay, while further meiosis first is classified into further four stages, that is prophase 1, metaphase 1, anaphase 1, telophase 1. But before going into all these phases, I want to tell you that meiosis first is actually a reductional division, while meiosis second is not a reductional division. Meiosis second is similar to mitosis. Okay, here you can see the homologous pair of chromosome gets separated. Homologous pair of homologous chromosome gets separated, while in meiosis second, the centromere of chromosome gets separated. Okay, so let's talk about prophase 1 in detail. Now let's come to prophase 1. So what happens during prophase? The chromatin thread, it starts condensation. Okay, and other things, the cell organelles starts disappearing, and the nuclear membrane gets it starts disintegrating, nucleolus starts disappearing. But it's in meiosis 1, prophase 1 is a very complex process. It undergoes several changes. And that changes are categorized in different phases, like liptotin, zygotin, pectine, diplotin, dikinesis. These all changes can be seen in chromatids, or you can say chromosome. Okay, these changes can be seen there. Okay, so these all changes are seen in nucleus. So let's talk about liptotin. What changes we see? What happens in prophase first? Liptotin, that is, this stage is characterized by the thin and longest chromosome. Thin and longest chromosome, this is very important. It has been asked, it has been already asked in previous year question. So this is characterized by thin and longest chromosome. While during the last of this leptotin stage, what happens? chromatin starts condensing, and chromosomes like a structure starts appearing, and this chromosome form a bouquet-like stage. It form bouquet-like stage towards centriole. Now see, suppose this is centriole, so what happens? It forms a bouquet-like stage like this towards centriole. Okay, so this is only the important thing to note from leptotene. Now let's move to other stage, that is zygote. This is very important. What happens? Two chromosomes, suppose this is a chromosome, this is a chromosome. What happens? These both chromosome comes together, starts coming together, and you will see that if they will come together, this will be called as synapses. This is called as synapses. This is called as synapses. Okay, so this synapses have two chromosome. This is called bivalent. This is called bivalent. While it also have tetrads. It also have tetrads. That is four chromatid. Look, it also have four chromatid. But I want to tell you that these chromatids are not clear here. These chromatids are not clear. Okay, you will experience these all four chromatids distinctly in the next stage, that is in pecitine. Okay, so what happens during zygotine? Two chromosomes starts coming closer. Okay, and you will also see some material between them. That material, suppose I want to show you this material, and that material is synaptonemal complex. That is called synaptonemal complex. Okay, so with the help of synaptonemal complex, these two chromosomes come closer. This is called synaptonemal complex. Okay, so pecitine is characterized by synapses or synapses, which is bivalent or tetrad. Synaptonemal complex is formed between both these two chromosomes. I want to tell you that liptotine and zygotine are short-lived. They are not very long stages. Okay, they are very short-lived. Okay, now let's move to other stage, that is pecitine. What happens in pecitine? Pecitine is characterized when these two chromosomes come very closer, and what happens? What happens? Their chromatids are now distinct. You can see their chromatid uh, very clearly. Okay, now they coil around each other. They coil around each other, and what happens? One somewhere on the chromatid of each of the chromosome, there is formation of crossing over nodule or recombination nodule. This is called recombination nodule, here or here, anywhere. Okay, so this is called recombination nodule, which appears on non-sister chromatid. Like suppose if this is chromatid, they both will be termed as sister chromatid. What about this chromatid and this chromatid? They will be termed as sister chromatid. But what about this and this? This will be termed as non-sister chromatid. Okay, so recombination nodule develop between non-sister chromatid, that is on suppose this blue and this pink one. Okay, now what happens? The genetic material gets exchanged here. That is, the blue part of this chromosome will get transferred to this pink one, and the pink will be transferred to this one. It all happens with the help of enzyme that is called recombinase. The enzyme is called recombinase. It includes two enzymes. Endonuclease, one is endonuclease, other is ligase. What happens? Endonuclease helps in incision of the chromatid or DNA or chromosome, while the ligase add them. Ligase help in joining them. Okay, so with the help of these two enzymes, what happens? The crossing over occurs. The exchange of their parts occurs during pecitine. It is a long process. So this is a very long stage. Pecitine is a long stage. Not very long, but this is a long stage. Okay. And you will see the four chromatids. So this is also called bivalent tetrad. Sorry, this is also called tetrad, and bivalent, obviously two chromosomes, so bivalent, you can say bivalent. Okay, so the crossing over or the exchange, that is same. It occurs during Peg-18. It's a in this stage the chromosome is thick. In this stage chromosome is thick. Okay, so note it. What can be noted from this Peg-18? That bivalent and tetrad is very distinct. Recombination nodule develop and crossing over takes place during Peg-18 with the help of recombinase enzyme, that is endonuclease plus ligase. That is it. That's it. Okay. Now let's move to Peg-18, from Peg-18 to diplotin. So what happens in diplotin? In diplotin, the chromosome, look, suppose this is chromosome, this is chromosome. What happens now? Some part is here and some part is here. That is recombination occurred. But they are not separated. They are not separated. They are still joined or connected with the help of synaptonymal complex. So what happens during this stage? This synaptonymal complex starts dissolving. Okay, and now they starts repelling each other and they move. But what happens where the crossing over occurs? It does not get dissolved. Okay, so one structure like crossing over, where the crossing over has occurs, now see, you will see this type of a structure here. This structure is called chiasmata. Okay, this is called chiasmata. You will see chiasmata here. Okay, because synaptonymal complex didn't get dissolved here. Okay, only this part synaptonymal complex is dissolved. So this acts like a structure is called chiasmata. So what to be noted from diplotin? From a neat point of view, I would like to tell you that in diplotin, synaptonymal complex starts dissolving. Okay, one. Other is the appearance of chiasmata, and also want to tell you that in some of the vertebrates, in some of the oocyte of vertebrates, diplotin remains for several years. Okay, diplotin remains for several years, so it is the longest stage. Diplotin is the longest stage, you can say. Okay, now let's take to other topic, that is dikinesis. So what is dikinesis? Dikinesis, here the synaptonymal complex get dissolved fully and the chiasmata open like a zip, in a zip like a manner, and two different types of chromosome appears because recombination is now completed. Now see what happens. So this type of a structure will appear because the recombination is already completed. Okay, and the prophase 1 will end by the disappearance of nucleolus. You know, in the prophase 1, nucleolus disappear, other cell organelles disappear, nuclear membrane disintegrate. So this thing will happen, and now the cell will move in next stage, that is metaphase 1. Now let's talk about metaphase 1. Metaphase 1 is typical like mitosis. All the cell will segregate, all the cell will no, all the cell will congregate. That is, all the cell will come at one plate like this. Sorry, all the chromosome will come at equatorial plate like this. Okay, centriole will move. In fact, centriole is already moved in the prophase first at the pole, and the spindle fiber from the centriole will get attached to homologous pair of chromosome. It will not get attached to same chromosome. Okay, it will get attached to different chromosomes. Okay, like in this manner. And in the anaphase, what happens? These two in the anaphase, sorry, it's telophase now. Okay, so what happens in the anaphase? Let me tell you here. In the anaphase, what happens? the chromosomes separates like this. Suppose this is chromosome, spindle here, spindle here. So this homologous pair of chromosomes separates in anaphase. So I want to tell you that in anaphase 1, what happens? Separation of homologous pair of chromosome occurs. Separation of homologous pair of chromosome occurs. Separation of homologous pair of chromosome occurs. After that, anaphase 1, what happens? Telophase. Telophase is just like vice versa of prophase of mitosis. So what happens during this phase? Cell organelles starts reappearing, nuclear membrane starts appearing, nucleolus starts appearing, and now in the cell you will see two nucleus. And just after telophase first, what happens? Cytokinesis will start. After the end of telophase 1, cytokinesis will start and the cell will be segregated, or cell, and the new cell, this type of cell will be formed. Suppose it was a parent cell. Suppose it was a parent cell with this type of scene. See, so at the end of the meiosis 1, two cell will be formed. This, I have told you that in anaphase 1, the chromosomes, homologous pair of chromosomes separates. So you will see like this. See like this. So what happens basically, if this chromosome is this, if this cell has 2n chromosome, if this cell has 2n chromosome, so this will be having n chromosome, n chromosome. If this cell has 4c DNA, this will be have 2c DNA, 2c DNA. In fact, what I am showing you here is the cell after S phase, because you can see the DNA is doubled. Okay, so 4c is for DNA and 2n is for chromosome, and we know that number of chromosome is calculated through the number of centromeres present. So here you will see one centromere, one centromere, so two centromere, so 2n. Here only one centromere, only one centromere, so n chromosome. Okay, so this is the termination of meiosis 1, of meiosis. Okay, now I want to tell you that meiosis second stage is 100% similar. Meiosis second is 100% similar to mitosis. 100% similar. So it is similar to mitosis, and at the last of this mitosis, that is meiosis second, what you will see in meiosis first, what to be noted, anaphase. During anaphase, homologous pair of chromosomes separates, but in meiosis second, centromere of one chromosome gets separated. That is, now see here the separation. This and this, let me show you with other color. This is the chromosome. So they both are separated because here the centromere, it splits. Okay, here the splitting of centromere occur, but here in the meiosis 1, in meiosis 1 what happens? Let me show you. In meiosis 1, these two are separated, but in meiosis 2, these two are separated. Okay, so this is called separation of chromatid. This is called separation of homologous pair of chromosome. This occurs in meiosis first, anaphase first. This occurs in meiosis second, anaphase second. And at the end of the meiosis second stage, you will see that 4 cell is formed, because one cell gives rise to two cell, one cell gives rise to two cell, and we have two cell at the end of the meiosis first, or the starting of the meiosis second. So at the end of the meiosis second or the meiosis, you will get 4 different cells with different type of chromosome. Okay, just because of recombination. Recombination is the reason for the variation in the sexually reproducing organism. Okay, which is very important. And meiosis is also very important because it restores, or it also what you say, it it maintains the ploidy in a species. Okay, so this was the important to be noted other from the need point of view. Okay.